on the palisade layer of the micellar pseudophage through the electrostatic and edge-bending interactions. Acknowledgement UGC for the financial support, and my collaborator, Professor P.K. Sains, my students Krishnendu, Anil, Professor Dr. Barik, and Professor Sanjay Bor, Jadavpur University. Thank you for your patience.
Thank you, Dr. Pal. If there are questions, only one or two questions. Thank you, Dr. Pal.
Okay.
Wonderfully. Thank you.
Thank you, sir.
Yeah.
Okay.
Yeah, please.
During your introduction, you said that the amino acids get oxidized by the catalyst gold 3.
It's not a catalyst, it's the oxidizing species. It's the oxidizing species, gold 3.
It gets reduced to gold 2.
Gold 3 to gold 1, finally. Gold 1 is the stable one. Gold 2 is unstable species. Sorry?
Some of the intermediates can be toxic.
Yeah, that's true. Gold 1 is also toxic.
So, gold 1 also is
Toxic.
toxic. So, what your basic objective was why you studied this reaction?
Yeah, that already I have mentioned that gold, some of the gold complexes, gold 3 complexes that are basically used as the metallo drugs for the anti-cancer and anti-tumor activity. So, because you know that when that gold drugs we have taken, so this gold 3 that can be reduced by the amino acids or proteins.
Process, some of the intermediates can be toxic.
Yeah, that's true.
So,
That's true.
not affect
Yeah, it will definitely affect.
adversely.
Yeah. So that mechanistic path we need to know. That is our main motto. Thank you, sir.
Thank you, Dr. Pal.
Okay.
Thank you, Dr. Pal. Next speaker is Mrs. Sangeeta Makone.
Now I request our chairperson, Dr. Professor Donke sir, to present a memento.
Constraint. Now the next speaker is Mrs. Sangeeta Makone. She is professor and ex-director of School of Chemical Sciences, SRTM University, Nanded. She has several publications to her credit. Then few students, some 7 students have been awarded PhD under her guidance. 4 students have been awarded MPhil under her able guidance. She is having the research projects funded by the national agencies. Then she has held number of responsible positions in her career. She has been the noted speaker also for some lectures, and she is having Women Scientist Award to her credit. And she has attended several and organized several seminars and conferences. So please welcome Mrs. Makone. Thank you very much. And
thank you. Good afternoon, one and all. Keeping in mind the time is very short, let us welcome all of you in this 108th Indian Science Congress for invited talk. On the title of my talk is related with one of the sustainable stream, that is nothing but the green chemistry related reusable materials, which is nothing but ionic liquids. So ionic liquid perspectives for synthesis of organic heterocyclic compound. One of the compounds we are considering here has a leading 4,1 derivative and evaluation of its anti-cancer activity. Actually, many more activities have been studied by my PhD scholars. So one of the work, you all know that what are ionic liquids, isn't it? Anion and ion combination in the form of liquid at the 100° near about temperature is considered as an ionic liquid, and it has a wide applicability as a solvent, as a catalyst, but we are considering here few perspectives. So in this brief out what does it mean. It is nothing but a combination of cation and anion. When we say it is a mixture of physical chemistry, inorganic chemistry and applications to the wide spread chemical sciences. So these are the common anions and cations. Generally inorganic and organic combinations are preferred. These are most of the easily used applications in organic synthesis of this ionic liquid, and we will start our work that in our research group they have synthesized new ionic liquids, and the motto of this synthesis is nothing but you are observing here very easy to synthesis with readily available material, and these materials are used to synthesize these five you can say ionic liquids, and in that particularly the acidic ionic liquids have been generated. There is a wide choice. Ionic liquid is having very fantastic physico-chemical properties. They are thermally stable. They are having high melting point and low boiling point. Even that is manageable. They are also called as designer solvents also. Future solvents are used as a fuel in the rockets. So many. Today only the one of the lectures regarding the biomass separation using this ionic liquid to form the eutectic mixture. So in case of environmental chemistry also, the time is I can't explain within 10 minutes even the application part. So if you consider this synthesis of this ionic liquid, that is nothing but the DACO-based ionic liquid. In short, now I will use the short form for this. DACO-based means di-anionic and di-cationic. Generally, the ionic liquid is a combination of monoform, one cation, one anion. But instead of that, two cations and two anions, this innovative form of ionic liquids we have synthesized here. And using this is the procedure regarding the synthesis. It is available in the publication. These papers have been published in catalysis letters and sulfur also. So these ionic liquids, both cation and anion, plays the role, and just like a tailor cuts the cloth and design the dress material and gives the color combination perfectly. So accordingly we can choose the cations and consider the organic functionality of the cations and anions to generate the catalyst as well as solvent. So many applications you can widely used for this. So these are the designer solvents as well as just like a cutting. You can cut paste these cations and anions. So these are the synthesized, newly synthesized, and their physico-chemical properties has been studied. One of the properties is nothing but moisture resistance property, which shows that it is having a thermal stability. These five, among these perchlorate DACO-based anions, ionic liquid, other anions, acidic part we have used halides, particularly using alkali halides, alkaline salts. These chloride, bromide and iodides are used to substitute these ions in the dicationic ionic liquid. So these are the newly synthesized ionic liquids. Weight has been considered and their absorbed moisture is counted, and comparatively it is found that these are diagrams for their absorbability, and it is found that these are thermally stable. This part is used for the seasonable synthesis. So these are not affecting by the temperature. So this has been considered for the further part of the synthesis of the organic application. So one of the application, these are the characterization of the perchlorate salts and ionic liquids. So identification of all these newly synthesized dicationic, dianionic, ionic liquids have been proven with the help of this. Then this is the spectra of another synthesized ionic liquid. So in this way, we have used the applications of this ionic liquid in the synthesis as a green protocol to develop an alternative for the synthesis of this thiazolidine 4,1 derivative, and its cytotoxic activity is also studied, and particularly the very prominent effect is found as an anti-cancerous compound for these sulfur derivatives. Here 6 different new 6 derivatives have been designed using this as a solvent and catalyst. So this has been studied, and this MTT assay against this MCF cell line has been observed. With the help of this, we got that it is nothing but this is the graphical abstract of our applications of ionic liquid for the synthesis of these heterocyclic compounds, sulfur-containing heterocyclic compounds. This is our present work. Simple procedure is there, simple schemes are there, and even in MSc project also we do using ionic liquid as a solvent, using ionic liquid as a catalyst. So many perspectives have been studied, considering the temperature variation, considering the amount of catalyst, considering the various solvent, and widely applications have been found. So this is about the result and discussion. It is found that all the synthesized dicatonic, anionic, DACO-based ionic liquids are acting as a good catalyst, which stoichiometric as well as minimizing the percentage in mole percentage. So this has been used to evaluate the appropriate amount of the catalyst. Catalytic study has been done using the model reaction, and the catalytic efficiency of this DACO-based ionic liquid was evaluated, and all the findings demonstrated that the newly synthesized ionic liquid promoted reaction in very short time with higher yield, near about above 80% yield has been occurred. Where the same study has been carried out using the monocatalytic ionic liquid. So there the yield is less and time is more. So this is about the observation, various moieties, and this is catalytic study and solvent study using various solvent and catalysts. This has been model reaction has been studied, and reaction conditions, appropriations of the reaction condition has been finalized, and using that, then this has been used in a particular. The beauty of using this catalyst, ionic liquid as a catalyst is there. That is why it is a green catalyst, because not 100%, but we are giving either alternative, greener alternative to the present methods, and these catalysts can be recycled for 5 runs, and this is the recyclable catalyst. So that is considered for the sustainable development for the synthesis of this various thiazolidine 1,4,4,1 derivatives using this newly synthesized ionic liquid. So there are various methods for this ionic liquids also developed. So this is the mechanism of this synthesis, and this is regarding the anti-cancerous compound. So out of the newly synthesized compound, few compounds they show the moderate anti-cancer activity, but 1 or 2 compounds which is having both the elements, that is the heteroatoms nitrogen and sulfur. Also the position of sulfur and nitrogen differs, then the anti-cancer activity varies. That also we got in the conclusion. So this is about the cytotoxic effect, cytotoxicity of that compound has been studied, and this is the percentage of cell viability against MCF cell and concentration that has been applied for the synthesized compounds. So I see 50 values for the test compound against MCF cell lines by MTT study after incubation period of 40 hours. It has been proven that these are the best anti-cancerous activity proven compounds, or it can be applied on wide range for the anti-cancers, particularly the breast cancer in the human beings. So these are the spectral data which proves the synthesis of these compounds. This is the conclusion. So doing this small piece of research work that has been considered that this investigation is eco-friendly, non-toxic, thermally stable, reusable ionic liquid as a catalyst in aqueous media. And this use of aqueous media is again helping to enhance the run of the ionic liquid, because the product is collected in the ethyl acetate or organic solvent by phase separation method, and the cytotoxicity of synthesized compound against the human breast cancer cell line, that is revealed that 5 tested compounds are effectively found to be anti-cancerous compounds. So at last, these are the publications out of this outcome of this research work, that is Journal of Sulfur Chemistry and Heterocyclic Letters and Catalysis Letters. So I am very much thankful, Honorable Vice Chancellor, for giving me this opportunity, first of all, to sanction DL to come out here. Dr. Uddhav Bhosle sir of our university, Director, School of Chemical Sciences, Dr. Santosh Devsarkar, and At this outset, I am very, very thankful for the Dr. Ranjana Agrawal, President of Chemical Sciences Section, giving me this opportunity to stand before you and present this small work in a beautiful way among these eminent persons gathering of academicians and scientists and teachers and administrators. So thank you and
Thank you, Dr. Makone. If there are few questions, 1 or 2 questions, she will be happy to answer. Any questions? So it seems that lecture was self-explanatory, and thank you very much. Thank you.
I request the chairperson, Professor Dongke sir, to present a memento to Dr. Sangeeta ma'am.
Dr. Sangeeta has been a product of our sister university, Amravati University. Thank you very much. The next talk is by Dr. G. Murthy. G. Murthy is there? Yeah. Just a minute. G. Murthy, in fact, he has not done only PhD, but he is DSc also. And then he has got DST Young Scientist Award. And then he is having other number of awards. He is a gold medalist also. And then he has worked for postdoctoral at several places. So I am not going to mention that. So he is having number of publications. His biodata is too big for me to read. So let us have Dr. Murthy with us. And currently, he is director of research and professor and head principal and scientist in charge of CEO, R&D principal investigator, project director. He is having several positions, and CEO and project director of Hamsa Research Foundation, Tumkur, Karnataka. So let us welcome Dr. Murthy. Dr. Murthy, please.
Yes. Thank you very much, sir. At the outset, I express my sincere thanks to Madam Ranjana Agrawal, sectional president of this prestigious Chemical Sciences Division, and the same is extended to Dr. S.S. Dongke sir for giving me this opportunity, and I am here to give a concept.
This is the pointer.
Oh, okay. It's a smart chemistry, and we have so many disciplines, starting from organic chemistry, inorganic chemistry, physical chemistry, analytical chemistry, environmental chemistry, green chemistry, everything is there. But it's a smart chemistry because taking into the phenomenon, the original phenomenon of chemistry, how we can go with energy. Because since 3 days, we went with so many concepts: agriculture, medicine, vaccine, and this is the energy related aspects. How the chemistry is having its pivotal role on energy, particularly bioenergy, that is bioethanol. Here I am focusing because we have done a lot of things on biodiesel, bioethanol, biogas, but it is a huge project under which I have taken a bit of this objective that is only related to bioethanol. Yes, you know Dalton's atomic theory? No, it is not Dalton's atomic theory. For your kind information, the recent investigation clearly elucidates its atomic theory given by Acharya Kashyapa. Acharya Kashyapa, he is the pioneer. And we say the father of chemistry, Antony Lavoisier. So that is also a conflicting aspect. So let us not discuss on this platform. Let us go with chemical characterization of fruit waste, sustainable sources, alternative feedstock for bioethanol production and performance analysis with the diesel engine system using gasoline and ethanol blends. Yes. The next slide. Energy scenario. Biofuel is nothing but any energy derived directly from either plants or animals or some primitive plants, algae, microorganism, biomass, or something sustainable waste which is being generated as a activity of human beings. So here we are going with bioethanol only, not biodiesel, bioethanol. Yeah, the flow of talk comprising this background of the research and where exactly is the bridging gap, because we are facing lot of things relating to the fossil fuels which is being deteriorated day by day, and by end of 2050, the fossil fuels will be depleted in a severe way. Hence, we are going with an alternative search of different bioresources. But amongst bioresources, again the cost-effective technology is amounts to be a greater interest. Hence, taking into consideration of these things, I am going with sustainable. It's a waste material. That a waste material can be subjected to develop or a protocol, a standard approach or line of attack to deliver the bioethanol at the end. Yes, baseline survey, then production of bioethanol, characterization, parametric analysis, techno-economic feasibility, that is cost-effective magnitude, and ultimately biofuel promotion to the village farmers. Then conclusion. Yeah, the massive energy demand. The demand is going every year. It is going higher, but the production of energy is going lower. That is the scenario where we are intersecting. That intersection is a negative phenomenon. Yes. So, as we know, the combustion of fossil fuels contributing mostly towards the environmental pollution by emitting various gases, ultimately emitting greenhouse gases, and is causing atmospheric pollution and ultimately global warming. Once it was a local problem, then it came into a global warming because of fossil fuels. Yes, next. It's not moving. Now it's. So, taking into consideration of such statistics, we are going with the search of alternative sources of energy. That is, global verdict was out for search for very alternative energy. Yes, as a strategic supply, energy is an important foundation for the development of economy and a society. With the growth of socio-economic and technological advancement in the past few decades, energy demand has increased considerably. Yes, next, this is a quick look on the current status of energy. See, there has been a continuous efforts by researchers who are strengthening the methods for utilization of renewable and green energy bioresources with sustainable approach, taking into consideration of different countries, and see the status of our India and the worldwide, the global energy demand. This is the pictorial approach. The demand is created. Yes, this is the graph showing, yes, energy demand is going higher up to the statistics of 2022, and the energy production is going down considerably. It's a negative approach. Yes, next thing is biofuel in the global perspective, taking into consideration of country-wise and the comprehensive production of biofuels in the global perspective. Taking the statistics from Clean Technica 2020, Brazil and United States lead the world in the production of biofuels for transportation. And other countries have also developed biofuel programs, including China, India, Canada, Thailand, Argentina, Australia, Colombia, respectively. Yes, this is the biofuel scenario, that is bioethanol production scenario. Very simple approach, very smart. Why I expressed in the beginning itself, smart chemistry means we are not dealing with regular chemistry, I mean basic chemistry, or something going with very complexity. If it is complexity, again the cost effective magnitude will not be satisfied. Hence I am going with smart chemistry. Yes, input material, fermentation, ethanol obtained, and distillation, and ultimately dehydration, giving 99.5% with purity of ethanol. Ethanol generated out of this waste material, the ethanol is very much similar to the ethyl alcohol obtained in the laboratory on regular approach. Yes, the objectives starts with a collection of bioresources and characterization, chemical analysis, and ultimately we are going with engine analysis. After having bioethanol out of this waste product, so ultimately we have to give a strategic conclusion, logic conclusion, then the transfer of technology to the beneficiaries or the industry, concerned industry. Yes, we are going with these two things. And materials methodology, baseline survey, starts collection, waste fruits. Yes, why waste fruits? I will tell you in the subsequent slides. The samples were subjected for analysis using standard procedures, And I am not dealing with any procedures here because already known to everything is known to you people. Fermentation via saccharification using baker's yeast was employed. Baker's yeast easily available. So I am taking only simple approach, not with a complex one. And the bioethanol, the objectives of the proposed study is implemented at the University Department of Chemistry, Ranchi University, Ranchi. Because since I have taken my DSc in that university, as an alumni, I am contributing this component to the betterment of the department. Yes, this is the experimental design and methodology. It's a flow diagram. Yes, biodiesel, biogas, bioethanol. We are going with only bioethanol. Yes, fruit waste collection, extraction, juice extraction, characterization, fermentation. Saccharification is nothing but yeast fermentation. Another word terminology, microbial terminology is called saccharification. Then purity test, that's GCMS also, and also ASTM standards. Yes, chemical analysis using CI, that is engine test rig, exclusively designed for ethanol fuel, IC engine, internal combustion engine. Then evaluation of emission parameters, NOx, carbon dioxide, SOx, oxides of sulfur, HC hydrocarbons, related pollution parameters, and developing a prototype, and the same has been transferred to the public domain. Yeah, this is the sources, because not only we went with fruit waste, and also non-edible fruit waste are also explored from the regions of Ranchi district, Kunti district and Pakur district. That's what in a bigger way I am projecting this, the geographical locations. Next, bioethanol chapter. So I am dealing with bioethanol chapter. This is only that fruit waste or the waste fruits. See the two terminology, fruit waste. After consumption, the waste will be generated. Every fruit will have some sort of generation of waste only. Or otherwise, if it is deteriorated or infected with microbes, that is waste fruits. So both are considered here, and it is rich in cellulose, semicellulose, and have a low lignin. Lignin will be very less, and this is beauty of science. So low, when it is low lignin, we can use it, the celluloses and hemicelluloses. In fact, fruits are rich in sugars. That sugar is converting into ethanol. Yes, every fruit generates 50% of its weight as a waste after consumption. After every consumption, 50% waste will be generated. And in addition, utilization of these fruit waste, bioethanol for bioethanol production may help in resolving the problem of waste management. Because in all local markets, including Nagpur, every corner of the country, if you go to fruit market or vegetable market, fruit waste will be available intensively. Yes, the bioethanol production from these sources may also help in carbon dioxide mitigation. Yeah, this is the sugar fermentation process. Very simple approach. That is biomass. Fruit biomass is pre-treated with acid or enzymes. Any suitable dilute, weak enzymes. Acetic acid. Weak acid. Acetic acid or hydrolytic enzyme. So in order to reduce the size of the feedstock and to open up the inner structure, that is plant cell structure, and using hydrolysis, that is hydrolytic enzyme, dilute acids and or dilute acids, so the cellulose and hemicellulose are broken down. Yes, ultimately the hydrolysis process breaks down the cellulosic part, fruit mass, corn to sugar, and ultimately the sugar with the influence of invertase released by yeast, glucose, fructose, and ultimately zymase will be converting into ethanol. Yes, the yeast contains enzyme called invertase and acting as a catalyst and helps in converting the sugar into ethanol. Yeah, yes sir, within 1 minute I'll finish. Yeah, this is a graphical abstract for your understanding, better understanding. Yes, the mixed waste fruits, pre-treatment, acid, either acid or alkali, juice extraction, filtration, mixed fruit juice sugar, and enzymatic hydrolysis, fermentation, distillation, ethanol, and CI engine interaction. Yeah, this is the data. So, bioethanol obtained from different waste fruits and along with mixed fruits. And here, different waste fruits and along with mixed fruits. So, reducing sugar analysis, this is bioethanol yield. And this is the picture showing the different waste fruits category and the mixed fruits. And these are the data. So, elemental composition, they are also contributing to the cellulose and hemicellulosic approach, ultimately leading to sugar, and sugar which leads in turn ethanol at the end. So this is the reaction again, and the properties of ethanol fuel, and this is 3 to 12%, average 3 to 12% yeast was subjected, glucose and simple sugars, ethanol and carbon dioxide ultimately. And see the engine parameters, and only 2 parameters I have taken here: total fuel consumption TFC versus BMEP, brake mean effective pressure, and it was reduced. The total fuel consumption was reduced, and specific fuel consumption, as a consequence, bioethanol showed a reducing effect on BSFC. this engine analysis, and this is carbon dioxide, carbon monoxide, oxides of sulfur, oxides of NOx, HC and fuel consumption. All the outputs with respect to data are significant to each other and while comparing with the fossil fuel. Yeah, outcome, that is mixed fruit waste can be recommended as a potential sustainable resource for yielding ethanol. Yeah, conclusion, ultimately, biofuels like bioethanol derived from sustainable biological sources are highly for sustainable development and are found to be promising, attractive, cost-effective and alternatives to petroleum fuels. Already, we have developed a CEM, cost-effective magnitude, and the same has been transferred to the public domain as well as concerned industries. And in addition, apart from the renewable qualities, they are reflected to play a key role in reducing negative environmental effects, that is fossil fuel products. Yes, this is the village program I have conducted to educate, to give awareness about how the fruit waste, biomass, agri-waste can be subjected for yielding bioethanol by taking into consideration of different village panchayats. And acknowledgement, University Department of Chemistry, Ranchi University, and for ultimately, thank you very much. This is energy, knowledge, Gnan, that is Daiva Gnan and Vignan. Jai Jawan, Jai Vignan, Daiva Gnan, Tantra Gnan. Thank you very much for giving this opportunity.
Thank you, Dr. Murthy. We can have one or two questions. Yeah.
Yeah, yes, sir.
Doctor, you are fundamentally only preparing ethanol
Ah.
from the waste.
Ah.
By any fermentation process, anything that contains sugar will give us ethanol.
Yeah, basic phenomenon, sir.
Have you blended it with some diesel or something to use as a fuel in the engine?
Blending is also possible, sir.
So you are burning
Yeah.
pure ethanol or blending? Because in the beaker, the color
That is ethanol only. Ethanol.
is ethanol, but it's not looking as if it is blended with diesel.
Yeah, blending and individual both are tried. But biodiesel blending ratio has been established. For bioethanol, alone bioethanol has been tried out here. And also we went with 5%, 10%, 15%. And the emission characteristic table is showing up 5% of blending, what is the status of emission parameters out of the engine interaction with bioethanol, 10%, 15%.
How does it affect the C10 number? Because engine is one thing.
The C10 number is very much similar.
You want to use it as a fuel.
Yes, sir.
I should know the C10 number or octane number.
Yes, sir.
Have you done that?
That octane number is very much similar to the ASTM standard. Very ASTM, American Standard Testing Methods.
Because a fuel, you don't need an engine to test the fuel. If you know the C10 number,
Yes, sir.
only I can use it in the engine. Otherwise,
That is understood, sir. Any fuel characteristic features admitting with the similar properties of ASTM, it is accomplished. But for a trial, we designed engines exclusively for bioethanol. Not diesel engine we used regularly, biodiesel using no.
you design the engine.
Yes. We prepared a test rig, engine test rig, exclusively for
But the
bioethanol.
must have directly used.
Existing, sir.
Can we have
thing-
this discussion afterwards?
Point number one, biodiesel engine directly you can utilize. In order to get the enhanced performance, bioethanol diesel with little modifications. Rest of the things are very much similar to diesel engine only, biodiesel. For bioethanol, we went with some smart modifications.
Thank you, Dr.
After
Murthy.
having all
in
these
fact,
things.
in fact, I also have one question, but I will speak to you afterwards.
Okay, sure, sir. Most welcome,
thank
sir. Otherwise, sir, you
Yeah.
take my email and I'll answer within 24 hours.
In fact,
More about interest, because all our students, they should know. See, you showed a pie diagram.
Yes, sir.
If you can show that India stands at third position.
Next, we start.
Consumption or something.
You can discuss.
The third, because
I will.
you see what happens, people blame us that we are the third or fourth largest emitter of carbon. But why don't you calculate per capita use of the fuel?
Yes, sir. For your
We are
information.
at 140th position.
Yes, sir. For your information.
Again, I request the chairperson,
It's a huge project.
Professor Donge sir, to present a memento to Dr. Murthy sir.
I'll communicate you. Thank you very much.
So just now we had a very informative lecture, and in fact we are short of time. I am sorry, Professor Enki, in fact, to interrupt you. I am extremely sorry for that, because we have to stick up to time. Now next lecture is by Dr. Aswar, but Dr. Aswar is busy somewhere, so he will deliver the lecture after some time. Is Dr. Monica Gupta is there? May I request Dr. Monica to come over here? Dr. Monica, she is from Jammu University, and she joined the Jammu University in 2010. And then she is also recipient of Young Scientist Award in 2012. Then Women Research Award in 2010. And she is having number of publications to her credit. And she has, in fact, she got first prize in oral presentation and several other awards. She has been awarded. She is a recognized supervisor for PhD, and 7 students have been awarded PhD under her able guidance. With these few words, I request Dr. Monica Gupta to deliver her talk.
This is Monica Gupta from...
Before I start, Dr. Monica, please stick up to time. Your time is 10 minutes.
I will try my best.
Yeah.
Namaskar, this is Monica Gupta from Department of Chemistry, University of Jammu. Today, the topic of my talk is nanomaterial are lead towards the sustainable development. It is presented in the Indian Science Congress on the topic Science and Technology for Sustainable Development with the Women Empowerment. Now, everybody knows that world starts from macro to micro. Macro means, for example, like grains of sand, which we can see with the naked eye. Micro means red blood cell, for example, which we require a microscope. For nano, we have a DNA, for example, for which we require a specially scanning electron microscope, which is SAM. So, if we have catalyst technology, these are divided into the 3 levels on macro scale, mini and micro scale. So, we have scale, we have purpose, we have material aspect. For scale, like macro level, we have overall conversion, performance, yield, productivity, lifetime, and for material aspect, we have catalyst, bed structure, packing matter, crust strength, and the attenuation. For mini and micro level, we have the effectiveness factor of individual reaction. For material aspect, we have the catalyst, particle shape or form. For nano level, we have intrinsic catalytic activity and selectivity, and for material aspect, we have catalyst composition, active structure or configuration. We have different type of materials like atom, protein, antibodies, virus, bacteria, eukaryotic cells, and corresponding to that, we have different nanomaterials. So this is recently 2022 Nobel Prize in Chemistry has been awarded jointly to the ACS Central Science Editor-in-Chief Carleon R. Bertozzi of Stanford University, Morten Meldal of the University of Copenhagen, and K. Barry Sharpless of the Scripps Research for the development of the click chemistry and bioorthogonal chemistry. So introduction to nanomaterials, it is a very basic introduction which was given by Norio Taniguchi, first used to term nanotechnology to refer to precise and accurate tolerance to minimize or required for the machining and finishing of the material. So there is a term like there is a plenty of room at the bottom. Before Norio Taniguchi, this term was coined by Sir Henning Friedman, and he was awarded Nobel Prize for this theory. So what are nanomaterials? These are having at least 1 nanometer dimension in the scale, and it involves an understanding and control of matter at 1 to 100 nanometer dimension, where it exhibits a unique phenomena leading to the novel properties. We have different type of molecules, water like nanobiomolecules, bacteria, cancer cell, pencil tip, tennis ball, and corresponding to that we have different type of nanostructures like fullerene, dendrimer, quantum dot, gold, metal oxide, liposomes, and polymers. So nanotechnology is an interdisciplinary field which has a wide importance in the area of chemistry, biotechnology, biology, medical sciences, engineering, and physics. So if we differentiate between nano and bulk materials, we have 3 basic categories. We have 3 basic points we can say, that is size, visualization, and examples. If we talk in terms of nanomaterials, which we can in the range of 1 to 100 nanometer range, at least in 1 dimension. If we talk in terms of bulk materials, greater than 100 nanometer in dimension. If we talk in terms of size, these are the materials which have the dimensions again in between 1 to 100 nanometer, and for bulk materials it is greater than 100. For visualization, nanomaterials which we can't see with the naked eye, for bulk nanomaterials which we can see with the naked eye. For example, like nanomaterials, we have different type of nanozymes, titanium dioxide, nanoparticles, graphene materials, and number of materials are available. For bulk materials, which we can like gravel, cement, sand, slag, and so on. So different type of nanostructures which are available like 0D clusters, that is the mesoporous carbon nanospheres, 1D nanotubes, fibers and rods. That is the, for example, we have the zinc oxide nanorods, nanotubes, fibers and rods which are in the 1D class. We have 2D films and cores like which we have hexagonal nanoplates, cubes-like structures. So we have 3D polycrystals which have a nanocubes-like structure. So different approaches which are used for the nanoparticle synthesis, top-down and bottom-up approach. In top-down approach, macrocrystalline material which is broken down and converted into the nanomaterial. While in bottom-up approach, it is a building up process where we have the formation of material from the basic structures. We have different type of bottom-up approaches, like we have sol gel method, we have chemical deposition method, we have the co-precipitation method, we have the sonication method. In top-down approach, we have a different type of method like sputtering method, we have laser ablation technique, we have micro assisted synthesis, and so on. This is a pictorial representation how the top-down approach converts a bulk material into the nanomaterial, and a bottom-up approach converts the atoms into the nanomaterials. So these are the different ways, different approaches which we can, different processes which we can use under the top-down and bottom-up approaches. And besides these, we have green synthesis under the class bottom-up approach, which include plants and microorganisms. Under plants, we have leaves, roots, flowers, and fruits, and under microorganisms, we have the bacteria, fungus, and algae. So everybody know the nanomaterials. They are widely distributed in nature, and nature is the best distributor of the nanomaterials due to their inherent properties, due to their remarkable properties. So these are some of the examples which are widely distributed in nature, like gecko's feet, which gets stuck on the wall due to the adhesive nanostructure present in the feet. Next is the nanomaterials which is available in the art and cultural heritage, like we have ruby red stained glass which gets illuminated due to the presence of nano-sized gold nanoparticles, like Augusta whose walls gets illuminated from inside and outside due to the presence of combination of 3 different type of nanomaterial like gold, copper, and silver. So application of nanomaterials are also distributed in the paints, in the drug delivery. Earlier in untargeted drug delivery was used, where the infected body gets targeted with the drug, and that destroyed the harmful cells besides the healthy cells. But nowadays we have a targeted drug delivery. Drug is tapped to the nanotube, it injected to the body, where it goes inside the body, it destroys the harmful cells, leaving behind the healthy cells and get released from the body. So we have in sunscreens, in cosmetics also, we have in construction. In construction we are using the silica. It is a part of the conventional concrete matrix. It imparts the mechanical properties or tremendously increase the mechanical properties of the mixture. In bridge production, titanium coated glass, they have the properties of anti-fouling and glaze. So it can also use in the energy. They have the capacity to store the huge amount of energy. Solar cells are also used in harder cutting tools also. So in high power magnets, due to the high surface area, in MRI techniques also. So now these are the reported examples which are nowadays already used in the day-to-day life. Now we have some valuable nanomaterials which we have synthesized in our laboratory, which has important application in the synthesis of biologically active heterocycles. Recycling of the waste materials into the nanomaterials is a step towards the sustainability. Now we are focusing on the sustainable environment, so researchers are also trying to use the waste material not only to minimize the waste material, but also minimize the cost of the waste material, but also to use the hazardous waste material and convert into the green synthesis. So this is the synthesis of adenine functionalized carbon nanocatalyst decorated with the zinc oxide nanoparticle. We have taken a cotton cloth, treated with the 1 monosodium hydroxide and 20% KOH, and stirred in the sulfuric acid, we will get the carbon structure. Now it is treated with the thionyl chloride in DMF, followed by refracting, we have the COCl, or the whole COH structure gets converted into the COCl functionalized carbon. Then it is treated with the adenine, it gets converted to the adenine functionalized carbon, followed by treatment with the zinc chloride in ammonium hydroxide. We have a zinc oxide nanoparticles get coated over the adenine functionalized carbon. These are some of the reactions which we have tried with the zinc oxide nanoparticles loaded over the adenine functionalized carbon. So recycling of the waste toner powder, it is one of the most important fact nowadays. The particle size of the particulate matter, it is of the size 18 to 16 micron. So it is of the so fine size, once it goes into the body, it can create the lung problem. So various efforts have been tried to convert this waste toner powder into the useful material. It is comprised of the carbon black, polyacrylate, polystyrene, ferrite and silica. So what we did, we have taken the waste toner powder, we have calcined at 600 degrees C, it converts to the ferrite, then it is treated with the ethanol and distilled water, sonicated for 20 minutes, then treated with the 28% ammonia anti-US, we have the silica coated ferrite. It is treated again with the amino propyl trimethoxysilane at 100 degrees C and reflects under the inert atmosphere. We have amino propyl functionalized silica coated ferrite. Then it is treated with the cobalt chloride hexahydrate in ethanol, stirred for 18 hours. We have cobalt nanoparticles loaded over the amino propyl functionalized silica coated ferrite. So these are characterized by the various techniques like HR10 micrograph of the functionalized catalyst. A and B structures indicate the layered structure, while the structure number C, it indicates the Sayed pattern, which indicates the polycrystalline rings are also present. Again, A and B on that side indicate the EDX presence a number
Dr.
of...
Monica, please conclude.
Yes, sir. Presents the number of elements present in the structure. So, these are again the comparison data which indicates that how in different steps it can be comparison. In the first step we have prepared the ferrite, in the second step we have prepared the ferrite over the silica, then we have prepared the complete catalyst, then it gets completely catalyzed and comparison will be done in all the different phases. So its application have been studied in the polyhydroquinoline derivatives and quinoline derivatives. So it has been tested for the various recyclable runs and found that this catalyst was found to be recyclable for various runs, and after carrying out the recyclability, we have taken again its HR time image which indicates the disruption of the catalytic structure, and Sayed pattern again indicates the concentric rings of the recycled catalyst. So in this way, these are the different type of catalysts which we have prepared: nickel ferrite over the boron, nitrogen, fluorine doped area. So these are applications have been studied in the pyrazolopyrimidine derivatives. So this is also studied for the various recyclable runs, and its XRD pattern even after the fifth run, it shows a fine peak in the intensity, and these are the various images. Sayed pattern indicate again the recyclability pattern after the fifth run and show that it is highly concentrated and it is highly polycrystalline ring structures. Yes.
Dr. Gupta, please conclude.
Yes, sir. I am moving towards the last slide. 56. It was 10 minutes, 20. In the end, I am also thankful to JKDST, DST GOI, Government of India, CSIR, UGC for providing me the fellowships, and I am also thankful to our Vice Chancellor, Professor Umesh Rai sir, for giving me a platform to carry out my research work. I am also thankful to the Ranjana Agarwal ma'am for giving me an opportunity to be part of this event.
Thank you, ma'am. Again, I request our chairperson to present a memento to ma'am. Thank you, ma'am.
Yes, Mummy is there now with you. Now today we are going to have in this session, the last lecture is by Dr. Mahesh B. Dr. Mahesh B is Professor in Chemistry and HOD of Chemistry in JSS Academy of Technical Education, Bengaluru. He has a teaching experience of more than 24 years. He is having number of publications to his credit. He is guiding students for their PhD degrees also, and he has delivered several lectures in national, international conferences. So I welcome Dr. Mahesh B and request him to deliver his lecture. And Dr. Mahesh B, please stick to timing, and your time is 10 minutes and your time starts now.
Thank you, Professor, for a nice introduction. Good afternoon. My field is synthesis of polypeptides and blending them with the commercially available polymers, aiming at the making of alternative wound healing materials. This is the abstract given in the book. We know peptides are not new materials. These were first synthesized in the year 1900s. By 1980s, the recombinant technology has come into exist, and many polymer molecules are obtained in the form of polypeptides. So the peptide-based materials are emerging as novel materials, especially for biomedical applications. From the polymer chemist point of view, I will discuss. Because most of the small molecules we use for medical applications, here I am talking on biomedical applications. Engineered biomolecules in protein purification, controlled drug delivery, tissue engineering, and biosurface engineering are being used. If you see this slide, I don't think the peptides are lagging in any of the fields. They are used in drug delivery, anti-cancer treatment, antimicrobial activity, anti-inflammatory, wound healing, cell culture scaffold, bioimaging, bioprinting, etc. So where do we get? We get peptides either from the nature or from the laboratory. From the nature, we get in the form of elastin, from tropoelastin, pig, soy protein, zine, collagen and gelatin, silk, as well as keratin. So they can be made as nanofibers, they can be made as films in the laboratory, they can be prepared as capsules, particles, mesoporous foams, hydrogels, microfibers, and nanofibers by various methods. Since my field is on tissue engineering, we are discussing on grafting the skin by using lab-made peptides. Synthetic peptides, compared to the natural polymers, in terms of peptides, synthetic polymers have more advantages. Reason is, you can have the required property by changing the amino acids. So if you change the amino acid, property of the polypeptide will also change. That is, we call it as tailor-made properties can be incorporated into the final polymer molecule. So that wherever the application is required, we can change the amino acid and we can produce the new materials. In our laboratory, we have made the polypeptide up to 250 amino acids, which are repeated in a polymer molecule. So 3 synthetic polypeptides we have made in our laboratory. They are elastin mimetic, plastic mimetic, and hydrogels. Elastin mimetic is the one of the polymer called tropoelastin, which has glycine, valine, proline repetitions, so that are being incorporated in a polymer molecule. On the other hand, plastic mimetic molecules, we instead of using glycine, valine, proline, we started with AVGVP molecule, which has elastic moduli and plastic properties. It can be heated to higher temperature and cooled to room temperature. They will retain the characteristic properties. That's why they are called plastic mimetic polymers. Hydrogels, they will have the properties of swelling and deswelling. So this can be utilized for tissue engineering applications. One such example we have made in our laboratory is poly-GGAP. So properties wise, if you come, very simple, but you know, raw materials are costly. That's why many people are not working like nanofield. Very few in India. Professor Govindraj group from Indian Institute of Science, SRM University, one group, and Amity University in northern part of India, they are working on this field. Main reason is the raw materials are costly. So, but properties wise, once you make the polymer, at room temperature, they are soluble in water, but when you raise the temperature slowly, they will turn into solids. This property is called hydrophobicity. and we call it as inverse transition temperature. Every individual polypeptide has inverse transition temperature, different inverse transition temperature. So you need not to go for big analytical techniques to characterize them. By knowing inverse transition temperature itself, one can characterize the polypeptides. So other advantages are biocompatible, biodegradable. Chemical method is costly, but if you use recombinant technology, then microbially, then we can get it at lower cost. So my field, I have been working from last 5 years in this. Actually, for about 11 years, I did not touch the test tubes, and I was not interested to do research because of I got afflicted with TB when I was completing my PhD. I thought that this chemistry is nonsense, I don't want. So, but later I realized that research is required if I want to survive in the any of the field. So I came back in 2017. I wrote a project to the Karnataka government. So they sanctioned me 35 lakhs to realize whatever I am presenting here. So elastin. Elastin is one such polymer which is present in, which is called as tropoelastin, which is present in the pig. So it can be produced in the laboratory. Professor Yuri group from Bioelastics Limited, USA, they have produced first this polymer. Later, Dr. D. Chennaiuda group from University of Mysore have done extensive work, and I am a student. So we have done extensive work in this field, and I continued the work from 2017 in this field. So elastin, you can see here, this the white portion, you can see that is the elastin which is present. It has alanine lysine rich alpha helical segments. It has both elastic as well as thermoplastic property. Design, simple. You take the general formula, VPGXG is the general formula. First you make VP, then GXG. GX stands for any amino acid. That amino acid we will keep on changing. Trimer, dimer, then we will combine them. We get the pentamer. Pentamer will be polymerized to get polypeptide. Chemical method we are using, of course, you can use recombinant DNA technology. So this is the simple, for students I'll explain. Two amino acids, you can subject them to condensation by using EDCA HOBT. In N-methyl morpholin, you get dipeptide. Similarly, D-block, the Bach group, by using ammonium formate and 10% palladium carbon, you get amine-free group. Then make it trimer. Similarly, dimer is ready. Similarly, trimer also you can make. Finally, pentamer, D-block everything. The TFA salt of polypentapeptide, it will be polymerized. So these are the polypeptides we obtained in our laboratory. Poly-GVGVP, poly-AVGVP, alanyl valine, glycine valine proline, poly-AEGVP, poly-GVGAP, poly-AVGAP. So polymerization. So you can do 1 molar salt solution you can take and polymerize it for 24 hours. So in DMSO, and then it will be done with or without HOBT and NMM as a base. First you get 3500 molecular weight cutoff dialysis molecule, then it can be obtained for 50 kD. So this is simple 1H NMR. This is carbon 13 NMR, which shows that polymerization has taken place. These are the polymers prepared and their corresponding inverse transition temperatures. We have made the blends. Actually, I'm not interested in synthesis of peptides. I want to make the blend with the naturally available polymers. So in this, the challenges are solubility of the polymers, possibility of denaturation because it's a sensitive molecule, selection of other commercially available polymers, knowledge for predicting miscibility characteristics.
Dr. Mahesh, please conclude.
Yeah. So 3 types of blends are there. These are the miscible blends, immiscible blends, and this one. So these are the methods we use to predict the miscibility of a polymer with other one. So we have published around 25 papers in last 4 years in this field. So these are some of the publications which evidences. So this is the recent paper. For the first time we got this electrospun material. Actually it was influenced by the mask which was made for the corona, this one. So IIT Delhi has made electrospun fiber for the mask. I used the same technology for obtaining polypeptide with polymer, a commercially available polymer. So this paper was published in a very good journal. So these are the applications we are trying. Wound healing applications mainly we are trying. I'm sorry, these are the materials we got in the laboratory. These are the papers published in the year 21-22. These are our students. Actually, in my lab, more ladies are there. All are married, so they are working in this field. And one student got awarded. This is my institute in Bangalore. Thank you.
So thank you, Professor Mahesh.
Thank you, sir.
Because of lack of time, there will not be any question answer session, but you can talk to him afterwards. So thank you, Dr. Mahesh.
I request the chairperson to present a moment.
Thank you, sir.
Just 1 minute. Friends, we had lectures from Dr. Biswajit Pal, then Dr. Murthy, Dr. Aswar is going to speak afterwards, Dr. Gupta and Dr. Mahesh B. So, and Dr. Makone. So she has left, it seems. I am thankful to the Indian Science Congress organizers for giving me an opportunity to chair one of the sessions. And I am thankful to my students, colleagues for giving me the wholehearted support. Thank you very much. Thank you.
Thank you, sir.
And I hand over the proceedings to Dr. Mahesh Anki to chair the session.
He was also former Vice Chancellor, G.S. Raisoni, University, Amravati. He was awarded by Maharashtra State Best Teacher Award 2012. He has 40 years of teaching and research experience. He has 34 students have been awarded PhD under his supervision. He has published more than 92 national and international publications in journal. Now, sir, please chair the session. Ma'am, to welcome Yankee sir by floral bouquet.
She did her PhD in 2008 from Kurukshetra University and has several publications to her credit and actively involved in research and development work. So I request Dr. Sumran to present her talk. Well, the time, again, we have time constraints, so please finish your talk within 10 minutes. Yeah. And we'll allow 1 or 2 questions.
Thank you very much, Dr. Yankee, Honorable Chair, and all the expert speakers, delegates. Very good afternoon to all of you. And at the outset, I would like to thank the organizer, ISCA, and my mentor, Professor Ranjana Agrawal, for giving me opportunity to present my work. I am presenting on topic iodobenzene diacetate mediated organic transformation, a sustainable approach towards aza heterocycles. The topic includes importance of aza heterocycles, advantage of iodobenzene diacetate, synthesis of quinoxylene N oxides, 1-aryl 4-methyl 1,2,4-triazolo 4,3-A quinoxylene, triazolo 4,3-A quinoxylene, 4 ons, 6-chloro 3-substituted triazolo 4,3-B pyridazine, 3,6 disubstituted bis 1,2,4-triazolo 4,3-B 3'4'F pyridazine by using green reagent iodobenzene diacetate. Nitrogen-containing heterocycles like pyrazole, imidazole, oxadiazole, quinoxaline, they constitute a broad class of compound that includes several successful drugs. Today, my talk is focused on the synthesis of aza heterocycles using hypervalent iodine reagent, that is IBD, because nowadays the researches are more being focused on the sustainable approaches, and the iodobenzene diacetate is a greener reagent because of several advantages. It is having unique, environmentally benign, mild selective oxidizing agent with reactivity pattern similar to heavy metal oxidant like salt of mercury, thallium and lead. Undergoes ligand exchange. Central iodine in IBD is fairly electrophilic. Experimentation involve very simple and neat procedure. So first series I am going to discuss is the synthesis of quinoxaline N oxide. Scanning of literature has revealed that quinoxaline N oxide are usually prepared by oxidation of parent heterocycle with various oxidants like hydrogen peroxide, acetic anhydride with peracetic acid, but the methods lack regioselectivity and results in formation of mixture of mono and di-N oxide. Other method involve the oxidation of benzyl alpha-aryl iminoxime with lead tetraacetate, but the toxicity associated with lead tetraacetate makes the procedure highly undesirable. So we explored the utility of IBD in this method. 1,2-dicarbonyl compound on reaction with hydroxylamine hydrochloride gives monoxime, which on condensation with aniline give Schiff's base, which on reaction with iodobenzene diacetate in DCM upon stirring afforded the quinoxaline N oxide. The mechanism involves the initial electrophilic attack of iodine on the oxime to give iodine 3 intermediate, which undergoes reductive loss of iodobenzene along with acetic acid to give nitroso derivative, which undergo electrophilic cyclization to give quinoxaline N oxide. The next series, we have synthesized various triazoles. The literature earlier reported method includes for synthesis by treating the hydrazinoquinoxaline with acid chloride and acetic anhydride, in which a red pigment is obtained by side product. Another method involved the reaction of chloroquinoxaline with mono and diacyl hydrazine, oxidation of hydrazone with bromine acetic acid or lead tetraacetate, or pyrolysis of hydrazones. Most of these methods suffers from several limitations like poor conversion, harsh reaction conditions, side product formation, high reaction temperature, tedious workup procedure, toxic and expensive reagent. So we explored IBD in this synthesis by reacting the hydrazine with various aromatic and heteroaromatic aldehyde. We synthesized hydrazones, and hydrazones on reacting with IBD under stirring in DCM provided the desired triazole. The mechanism of this IBD-mediated transformation involved initial electrophilic attack of IBD on the terminal nitrogen of hydrazone to give iodine 3 intermediate, which undergoes reductive loss of iodobenzene along with the expulsion of acetic acid to give nitrilamine intermediate, and final ring closure through nitrogen of quinoxaline ring give the diffused triazole. The interaction between small molecule and DNA is very important in molecular chemistry and medicinal research. The small molecule interact with DNA may act as groove binder or may act as intercalator or photocleavage agent. Photocleavage agent or photonucleases are the molecules which cleaves the DNA upon excitation. They start a series of chemical changes which ultimately lead to DNA cleavage with site selectivity or without site selectivity. And our compounds exhibited DNA photocleavage activity. When there is a breakage in strand, it leads to the relaxed circular DNA, which is called form 2, and the two form are easily separated by gel electrophoresis. Prompted by this preliminary result, which indicated that the photocleavage activity was dependent on the substrate as well as concentration, we designed the new fused triazole by replacing the methyl group at position 4 with the oxo group by using the same strategy, preparing first hydrazine, condensing with aldehyde to form hydrazone, and then oxidative cyclization with IBD. Here we have studied the mechanistic studies, revealed that we performed mechanistic investigation by using various additives as potential inhibitors and activators like L-histidine as singlet oxygen quencher, D2O singlet oxygen enhancer, hydroxide radical scavenger, mannitol, sodium benzoate, potassium iodide, and superoxide anion scavenger dithiothritol. By using these additives, we have determined that the mechanism involved superoxide anion radical formation and type 1 pathway is involved, and the reactive oxygen species, that is superoxide anion radical, is the involved in the DNA photocleavage. And the photocleavage, our compound show photocleavage, and this activity was dependent on substrate concentration and time of photoirradiation. Next, we explored the synthesis of triazolo-4,3-B pyridazine and bis-triazolo-pyridazine. The starting material was 3,6-dichloropyridazine, which on refluxing with hydrazine hydrate in tertiary butanol afforded the corresponding 3-hydrazino-6-chloropyridazine, which undergo condensation with aldehydes to give hydrazone, and on oxidative cyclization with IBD, we get the monotriazole, and by following the same procedure, we get bis-triazole. An interesting pattern was observed in the 1H and C13 NMR of these bis-triazole. Particularly in case of methoxy derivative, the spectra expected peaks were found to be exactly half. Pyridazine proton as well as aromatic proton were also half in number. So we decided to explore this structure of bis-triazole by doing the X-ray diffraction studies. X-ray diffraction studies revealed that in case of the substituent, where substituent is orthofluoro and parafluoro, it indicates that the molecule is not planar. It is having, because of twisting of these aryl rings takes place to avoid the steric hindrance, and the molecule crystallized in orthorhombic P2121 space group. So these are the X-ray structure of compound having fluoro substituent. And we evaluated the anti-cancer activity of monotriazole, that is triazolopyridazine, against 3 human cancer cell line, SBOL, NALM6 and an MCF breast cancer line. And our 3 compounds having 4-chloro, 4-bromo and indole, they exhibited the potential cytotoxic activity, and this compound was most potent. And the mechanism involved caspase activation in NAM cells. And particularly, SAR revealed that para substitution is more effective as compared to ortho and meta. Bis-triazole were also evaluated against HeLa cell line, but because of poor solubility of bis-triazole in the buffer medium, they are not evaluated. Only 4 compounds were evaluated, but these have low cytotoxicity. So, in nutshell, I would like to conclude that IBD is a versatile green reagent for the synthesis of aza-heterocycles. IBD offers effective, convenient, ecologically facile approach for the synthesis of quinoxylene, N-oxide and triazole. So, thank you very much for patient hearing.
It's open for questions, for discussion.
If there are no questions, let us thank the speaker.
Thank you, ma'am.
Dr. Sumra, for finishing it well within the time. On behalf of the organizer, I would like to present a memento to Dr. Sumra.
Dr. Ravi Kumar sir, whether he is present here? Dr. Ravi Kumar sir. Okay. Sir, we will next.
Yeah, we'll go ahead with the next speaker. I invite Dr. Ashok Kumar Jha from TM Bhagalpur University, Bhagalpur. His work, Dr. Jha is there. Welcome, sir. He's working in the University Department of Chemistry. He has had a meritorious career right from his schooling, graduation to post-graduation days, has several merit scholarships to his credit, and more than 50 research articles in different national and international journals. Dr. Ashok Kumar Jha will be talking on characterization and adsorption studies of bentonite for heavy metal removal.
Respected chairperson and the sectional president Professor Ranjana Agrawal for giving me an opportunity to speak something over my research. And actually, I have been working on the adsorption studies of bentonite mineral for heavy metal removal. And it would be in the fitness of things here to mention here that I have completed 2 major research projects sponsored by UGC on this research. Actually, this is the abstract. Now I am skipping it. So, actually hexavalent chromium and arsenic in drinking water has attracted the attention of researchers worldwide. And what we have done, that we have collected the bentonite minerals from Rajmahal Hill, and we have analyzed and characterized the bentonite mineral for different activities. Actually, the bentonite minerals are smectite group of minerals, and it has a layer structure, and due to the layer structure, the bentonite mineral has an excellent intercalation property. Means this can be modified by suitable reagent. This can be intercalated by suitable intercalator, and due to the exchangeable cations, it has a great property of adsorption as well as the exchange of ion, which makes it suitable for heavy metal remediation. So you see here, these are the components: silica, potassium oxide, alumina, and this is the cation exchange capacity, and K is the thermodynamic equilibrium constant here. The exchange property of bentonites has novel application in softening of hard water, and it has a number of industrial uses like the softening of hard water in wine industry, decontamination of radioactive waste, refining of oil and grilling mud in oil wells, and the industries are exploiting this vast resources of bentonite in a natural way in all the states. And it would also be proper here to mention that the Rajmahal bentonite, the Rajmahal hills have a very good treasure of bentonite minerals, and these are, earlier they were treated as low quality bentonite, but researches have proved that the Rajmahal hill bentonites are also good quality bentonites, and since then researches have gained momentum on this particular topic. So these are the materials and method. So actually what we have did that Jharkhand samples are typically Montmorillonite. Actually Montmorillonite unit is present in bentonite, and that is a phylloaluminium silicate and smectite group of mineral, and these minerals, this smectite group has been confirmed, has been characterized by TGA DSC study of collected samples. And when we have done the TGA analysis of the collected samples, we have found that the weight loss is around 14.4953%, which is very similar to the bentonites formed in USA and Japan. The earlier, they are very well in agreement with the experimental values having been obtained in Japan and USA. So these studies confirm that our bentonites are a good quality bentonite, and the weight losses are due to the loss of molecular water from exchange layer, the dehydroxylation of water. The dehydroxylation has also takes place, and next, so these can be these are the PXRD of different samples collected by me. This is the PXRD of Rajmahal Hill bentonite 5, Rajmahal Hill bentonite 6, Rajmahal Hill bentonite 8, 9. These are the XRD graphs, RB10 we have collected, and this is the FTIR of Rajmahal Hills bentonite. These are the peaks. The peaks are around 3600 cm inverse, which is show the presence of hydroxyl layer on the surface of the Montmorillonite, which gives it an excellent property of exchange or adsorption. This is in the same way, this is the FTIR peak of other sample, RB9, this is the, yes, here you see the weight loss is, the total weight loss is 19.216%, and the increased weight loss is due to the presence of some of the impurity. So first weight loss you can see in this graph like this here, and the total weight loss is 14.4953%. And here also this is the weight loss, and this is the DSC, the curve of the sample of RB9, RB10, and these are all the weight losses, 3 weight losses, which accounts for the total weight loss. And this is a comparison of TGA curves of samples 7, 8, 9, 10. You can see in the graph RB7, 8, 9, and 10. And this is the actually DTA curve of samples 7, 8, 9, 10. And lastly, what we have done, we have taken the bentonite sample, we have taken the initial concentration of chromium ion at 2 ppm, and we have measured the residual concentration of hexavalent chromium in 1 hour, in 2 hours, and in 3 hours. And after the treatment for different time intervals, we have calculated the percentage removal, and from percentage removal, we have calculated QT, CT, log QT, log CT, CT by QT. Actually, a graph of log QT versus log CT and a graph of CT by QT versus CT gives Friendlich and Langmuir isotherms respectively. And in the other table, we have done percentage chromium-6 removal after treatment with 1 gram bentonite for 2 hours, and the values of QT, CT, log QT and log CT have been calculated here. And we have worked with 2 types of 2 common isotherms, Friendlich and Langmuir isotherms, which is very common. Friendlich isotherm refers to the multilayer adsorption, as well as Langmuir to monolayer adsorption, and we are also working on Ilovich isotherm and other isotherms with the same experiment. So this is the result and discussion. With other samples, we have done these experiments with manganese also. This is the percentage removal of manganese, and manganese 7 removal, the maximum percentage removal is 7.25%. And here also we have increased the mass of the bentonite minerals. We have treated it 1 gram, 2 gram, and 3 gram. So this is the graph log QT versus log CT, Friendlich isotherm, chromium-6 ion bentonite system at 26 degrees C. Here also we have it for manganese 7 system. For different samples, you can see different colors for different samples. This is Friendlich isotherm. Here we have CT by QT versus CT. For Langmuir isotherm, manganese 7 bentonite system. And here for adsorbed amount of chromium-6 ion with time, the adsorbed amount of chromium ion versus time. And actually we have observed that this follows a first order kinetics, and from thermodynamic consideration we have come to the conclusion that a spontaneous adsorption is taking place because we have calculated change in Gibbs free energy. Change in Gibbs free energy is negative. It is accompanied by that. So this is the same images of bentonite before adsorption and after adsorption at 5 kx, and lastly the result, and same images, grains shown images resemble stacks of layers and aggregates. A highly compact structure formed by small curved or bent layers provide large surface area, and it has actually 2:1 dioctahedral structure, means one octahedral structure is sandwiched between the 2 silica, and same images before and after adsorption in figures clearly explain removal of chromium-6 and manganese 7. So these are the references. And lastly, I have published some of the good papers on my work, and I acknowledge the fund released by, sanctioned by UGC for my project. And lastly, I want to also pay respect to my favorite teacher, Professor N.K. Jha, former professor and head IIT New Delhi, who has actually inspired me to carry out this work, because he has earlier worked on this topic to some extent, and he advised me to go forward with this piece of work. So I have a lot of thanks, or express my gratitude to my teacher, Professor N.K. Jha. Thank you.
Thank you, Dr. Jha. Any questions, please? Well, bentonite clay, I have a question. Bentonite clay, basically the process is ion exchange or adsorption?
Okay, this is a very nice question. Actually, what happens? Both processes are taking place. In mesopause, in bentonites, we have calculated the surface area from nitrogen adsorption and desorption. And we have found that mesopores are there. We have calculated the surface area.
Okay,
so one adsorption is taking place due to surface area, but the ion exchange, cation exchange is also taking place. So both the properties have made bentonite an excellent ion exchanger as well as an
Adsorption, yeah. Well, this technology of adsorption, your chromium and these unwanted ions, from water you removed, now they are on the surface of bentonite. What should we do with that bentonite? Because now it is secondary pollution.
Yes, sir. It is a nice question. Actually, I also really feel it. But now we are working. What can be done? And actually, we have recently sent a project to the SIR, DST SIR, for working on the catalytic activity. And in that, we have proposed a plan to recover the catalyst after removing it.
Okay, so thank you. Well, this is something which all researchers, I would like to think, that try to find a total solution. No, we not like transferring it from one phase to another. The problem does exist somewhere else. It should not be that I have removed the problem from here and it's somewhere else. So work something where you are getting a total solution. Thank you once again.
Thank you, sir. I request the chairperson, Dr. Yenki sir, to present a memento to Professor Ashok Kumar Jha.
Thank you. Congratulations, Dr. So, now I would like to invite the next speaker, Dr. Rajdeep Dey. Dr. Rajdeep Dey is currently working as an assistant professor in Department of Chemistry in Sister Nivedita University, Kolkata, West Bengal. He has been awarded as a Fellow of Indian Chemical Society 2019, has 15 publications in reputed international scientific journals, and is actively involved in research and development. I request Dr. Dey to deliver his talk.
Thank you. Thank you very much for inviting me here in the Science Congress platform. And also I would like to thank Professor Yenki to give me the opportunity. So my talk is entitled with the novel low-cost green synthesis of water-soluble carbon nanoparticle. So first of all, why low cost? That is the very important here, because why the work is unique, we need to know. Because we have started the work with very popular, low cost and easy available material. And also to characterize those material is very easy. One can start with having a very minimum equipment, minimum infrastructural facility, because nowadays the infrastructural facility is a very big problem, because all the newborn universities are there, so they don't have a lot of facilities over there. So if we can minimize these characterization facilities, then it will be nice for us. So that's why I have taken jackfruit jackfruit, betel nut and guava all are very, very common tree in our societies, and we are just taking the bark of that three plants and we have continuing our nanomaterial and the carbonized product. But first of things, that why carbon? First we need to know that carbon is a very benign element from the very beginning of life, and all the time we are generally associated with the carbon and with the hydrogen, because it's a hydrocarbon, and which is a very important organic stuff for our daily life. But my story is different. My story is with carbon without hydrogen. I am not interested with carbon with hydrogen, unfortunately. So what is the carbon without hydrogen? It's a very simple metamorphism of the green plant. We know all these things, and we can get the coal graphite in material, and right now we are getting some allotropes carbon graphene materials, the carbon nanotubes, etc. So we can start with these things. And how we can get the soot, the normal carbon graphene soot, is very easy. We in the winter time we are seeing in our house the furnaces are there. We can collect the soot from here. In all our houses, lords are generally enchanted with this lamp, and we can collect the shoots from there. So it is very easy to collect. So based on that application, or based on that idea, I have started the same thing with the jackfruit, betel nut and guava. What we have done, that the tree of the tree bark of these three plants I have taken, and then I just I just washed it thoroughly with water and some pigment to ensure the removal of the lignin and wax material over that tree plant. And after that, the carbonize that material with a earthen pot. And this is also very very usual earthen pot. We can get it from the sweet shop, and with some lacquer we can arrange that lacquer, and we can just heat it this earthen pot with 45 minutes, and I will get that carbonized product. So this is very easy. No need to do any good or any big infrastructural facilities over there. What else? Then after that air dried soot, what we are collecting from that earthen pot, we can acidify these things several times. The same chronology, jackfruit, betel nut and guava. And after certain process of steps, the acid free stuffs are generally purified. Why the purification is required? Because there are some metal free catalysts are there, maybe there are some impurities are there, so it is required to free that catalyst and dry it on the water bath, and we are getting this dry product of these things. So these are the nanomaterial we have already prepared with very easily. But now it is very important that what are the application of that nanomaterial, whether it is the water soluble or not. Because if it is not water soluble, we can't use in the agricultural land, we can't use in the applied field. So we have checked with a very usual method, that is the acid based stoichiometric method, to check the carboxylic acid assay, and we found that 22% by weight such heavy carboxylations are there. There are the 5 several vials. Vial 1 just dropped the sample, and after 20 seconds, 40 seconds, and after 5 minutes we have seen, this is the last vial, we have seen that it is completely soluble with the water and without any sonication. And we have checked after several months also that it has no precipitation. So that means this is a completely soluble in the water system. So that is the very important part here. If it is not water soluble, then we can't use in the agricultural land. So now the next thing, the application. Where we applied, again I have taken very very useful, very common gram seeds. It is available in every houses, I guess. So we have taken and we just put the gram seeds on water for a day, and it can germinate it with a day, and I put it in 3 different vials, and one is for the blank. And I checked after 10 days, I have seen the jackfruit, the first one, jackfruit is growing much higher, the betel nut is somehow moderate, and the guava is very higher positions. So these are the basic things we are using here, that the gram plants growth very easily, the gram plant grows very easily, but the blank portions, there are no such growth of my blank nanocarbon materials. We have done some, definitely we have done some characterizations process. We have done the TEM analysis and powder XRD to check whether it has any changes with the temperature or not. And we have done the FTIR system to check the carboxylic acid. This is the OH and this is the C double bond O group. Carboxylic acids are present here. And we have done the Raman spectra also. That Raman spectra is also characterized that there are some degeneracy disorder and the graphitic modes are there. Now the question will come another thing, that is it toxic or non-toxic? Because if it is toxic, again we can't use in the agricultural field, because we have already done that it is the water soluble. So to check the toxicity, this is very useful method. Already we have done from the several times from the Mesolithic ages of life. We know that when the people wants to do the firing system, they know that how to do the carbon material, and nowadays we are doing barbecue in our picnic and we are getting all these things, and the chapati also, all are the nanocarbon. And I have collected these suits. Oh yes, the sizzler, the very important part. The sizzler, whatever we are using in the tandoori chicken and tandoori pakoda paneer. So that are also the carbonized product, and I have collected that carbonized product and I have checked, and the roti and the tandoori what we are getting in conference every day in the morning and daytime. So these all are the nanocarbon we are eating. So if we can eat, so it make no sense. So it is a non-toxic element, but still is a charcoal facial, nowadays charcoal ice cream, is a roasted corn, or all are nanocarbon. So we are using everyday daily life in our nanocarbon materials. So these are non-toxic, but still we need to check something with the assay. We have done some MTT assay, but for the publication purpose I have not given the data here. And this is also the same things that from the Mahabharata Ramayana ages we are using kajal in our eyes. So all are nanocarbon. So this is not toxic. So in summarize, we can say that this is a very quick, simple method and for the water soluble nanomaterial, to investigate the water soluble nanomaterial with the plant cell. But what is our future study? We have checked only in the gram plant system, but we need to check in the flowering system and fruit ripening system if it happens or not. So that is our future future studies of the things. And I must acknowledge to my university, Sister Nivedita University, and my collaborator BBAU University Lucknow. And thank you. Thank you so much.
Thank you, Dr. Day. Any questions from the audience side? Yes. Professor Tan.
Yeah, thank you.
I could understand the term water soluble, because when you say that carbon is soluble, I mean, I could not understand the terminology. How this carbon without hydrogen can be solubilized?
No, not the carbon. This is a carbonized product. That is a nanomaterial. This is already with the...
This product is something apart from carbon.
Exactly.
So carbon with some...
Exactly. Basically carbon will oxidized with the HNO3.
Some hydrogen.
No, not required, because a carbon material can be carbonized. When it will get carbonized, it has some carboxylic acids, say that type of things. Directly, I am not using the hydrocarbon material. So I told that I will not use the hydrogen as hydrocarbon. Yeah. Thank you.
Well, so let us thank the speaker. Thank you, Doctor.
Thank you, sir. Now I request the chairperson to present a memento to Rajdeep, sir.
Thank you, sir. Thank you. Thank you, sir. Thank you.
Next speaker, Mr. Dipender Kaushik, is he there? I think Dr. Rahan Dale is also not there. Dr. Asha Jain. Is Dr. Asha? Okay. So I request you to give a brief introduction of yourself and then give. Yeah, she is giving an oral presentation.
Continuing my research under supervision of Professor Asha Jain, Department of Chemistry, University of Rajasthan, Jaipur. The topic of my today's presentation is facile incorporation of zirconium-4 into aminophenol, substituted aminophenols and flexible N-thalloyl amino acids. Their spectroscopic characterization, DFT-assisted optimized molecular structure and Mulliken charge distribution analysis. As an introduction part, we have reviewed the literature and we have seen that the field of hybrid complexes of zirconium-4 has received tremendous attention in the past few years because of their wide variety of applications in the field of pharmaceuticals and technological fields. The complexes of zirconium-4 have been used as antibacterial agents, antifungal agents, antioxidants, and some of them are also used as anti-cancer agents. In technological fields, they are used as catalysts, as photoluminescence materials. A large number of reactions of zirconium-4 have been reported in literature with a variety of organic ligands. Mainly 3 metal precursors have been used for their synthesis: zirconium tetrachloride, zirconium isopropoxide, and zirconosine dichloride. I have used zirconium tetrachloride for the synthesis of new organic derivatives of zirconium-4. The type of ligands that are used and reported in literature to modify the reactivity of zirconium-4 involves Schiff bases, orthopentylated compounds, heterocyclic beta diketones, fluorinated, non-fluorinated beta-diketones, N-protected amino acids, and other ligands. Before starting our experiment, since we have performed all our reactions in strict anhydrous conditions, so we have dried all the solvents required using standard procedures. In experimental conditions, firstly we have synthesized the sodium salts of ligands. I have used 2 ligands, aminophenol, substituted aminophenols, and N-protected amino acids. And after the synthesis of their sodium salts, we have added metal precursor zirconium tetrachloride. NaCl was formed as the byproduct and it was removed. Then excess solvent was removed under reduced pressure, and finally we received newly generated dark colored solid complexes. They were dried and recrystallized for purification. Result and discussion. This is my reaction scheme. In this I have reacted zirconium tetrachloride with 1 mole of sodium salt of aminophenol, substituted aminophenol, and 2 moles of N-protected amino acids. Then we have refluxed this reaction content for about 6 to 7 hours, and finally we received this hepta-coordinated zirconosine complex. I have synthesized 9 complexes of this series using different variations of aminophenol, substituted aminophenols, and N-protected amino acids. I have used 3 amino acids, beta-alanine, leucine, and isoleucine in this. Then we have characterized these complexes, these newly synthesized complexes, with the help of spectroscopic techniques, with the help of IR spectra, proton NMR spectroscopic, C13 NMR spectroscopic data. As we can see in proton NMR spectroscopic data, proton NMR spectra of these complexes, the peaks for OH and COH group of ligands was found to be absent, which shows the deprotonation of these ligands and formation of zirconium-oxygen bond in these complexes. On the basis of mass spectrum of 1 representative complex was recorded, and in mass spectra we have not observed any peaks for the dimeric structure. So we have proposed this hepta-coordinated monomeric structure for these newly synthesized complexes. Some of these representative complexes were also studied with the help of computational techniques. We have used DFT B3LYP functional in Spartan 20 suite of computational chemistry for the theoretical investigation of 3 representative compounds, compound 1, 2, and compound 7. These are the optimized bond length and bond angles data of these compounds. We have also optimized energy, E homo, E LUMO, delta E dipole moment of these representative compounds. As we can see, these optimized ground state energies of these complexes can also be correlated with their stability. Since on the basis of this energy, we have concluded that out of these 3 complexes, complex 2 have lowest optimized ground state energy, which suggested that complex 2 is most stable out of these 3 complexes. Then the homo-LUMO transition energy gap may also be considered as the measure of reactivity of these complexes. Lower this energy gap, higher will be the reactivity. Then on the basis of Mulliken charge distribution analysis, as we can see from the bar diagram, that zirconium atom has higher positive atomic charge, and all the other atoms surrounding zirconium have negative charge. So this indicated the presence of ionic character in these bonds. These are the optimized structures of these representative complexes. In conclusion, we have synthesized these hybrid complexes of zirconium-4, and then we have optimized their molecular structure, optimized energies, E-HOMO, E-LUMO, and other parameters. These complexes may also be tested for their possible antibacterial and antifungal activities. These are the list of references. And acknowledgement, I would like to express my sincere thanks to Dr. Asha Jain, my guide, and Professor Sanjeev Saxena sir, to CSIR, and to Indian Science Congress Association for giving me this opportunity. Thank you for your attention.
Thank you, Ms. Soni. Any questions?
आपने zirconium का कौन सा salt use किया?
Zirconium tetrachloride, sir.
It will stable?
Sir, we have used strict anhydrous conditions, so usko open.
In case of zirconium complexes, sometimes it is very difficult to isolate.
Sir, if you use zirconium isopropoxide, then it is quite difficult to isolate and characterize. But zirconium, the complexes we
Complex is hexacoordinated or
heptacoordinated, sir.
And how does the valency get satisfied? Zirconium. Valency of zirconium.
Zirconium 4, we have used zirconium 4, so one chlorine atom and three OH groups.
Well, Professor Aswar, I would, because of the paucity of time, I have to interrupt. You may discuss with her. See, the next.
Okay.
So, thank you very much.
Thank you, sir.
Now I invite the next oral presentation from Dr. Suresh Kumar of Kurukshetra University.
My topic of my presentation is hybrid urons designing synthesis and biological evaluations. Now, based on the molecular hybridization, in molecular hybridizations, we use two independent pharmacophore and club them in a single unit to have some potential biological activities. And based on that, we have developed some hybrid type of urons. Uron is basically a subfamily of flavonoids, and it is responsible for yellow pigmentation to the various plant parts. This is containing benzofuranone nucleus, 2-aryladine benzofuranone nucleus. These are some potential biological activities which have been reported for the urons. Now, these are the reported routes for the synthesis of these urons. Generally, the best two methods are oxidative cyclization of these 2-deshydroxychalcones, and the second one is condensation of aryl aldehyde to these phenylcumulenones, or these type of moieties. And we have developed a sustainable approach for the synthesis of urons that we have published in Green Chemistry Letters and Reviews. And this involves direct cyclization of this phenesyl chloride with the aromatic aldehydes, and it is avoiding one step basically. It is one pot synthesis, and based on this synthesis we have developed certain hybrid types of novel urons. In first we clubbed pyrazole, this unit, to the benzofuranone units, and using the similar same protocols which we have earlier developed in our lab, we synthesized this different library of compounds, and then we evaluated these compounds for the anti-cancer activity against MCF cell lines. And see this percentage cell viability. If we compare with our compound with the paclitaxel, a reference compound, we found that most of the compounds, they have cytotoxic activity comparable to the reference compound paclitaxels. And see this paclitaxel is having IC50 value 18.5, and some of the compounds, they are having IC50 even better than the paclitaxels. These are the potent compounds which we have reported. After clubbing pyrazoles, then we went for the coumarin, another potential pharmacophones. We reviewed the literature and then we published the review in Chemistry Select, and based on that review, we synthesized some coumarin club benzofuranone nucleus, again using the similar protocols, and this library of compounds we have synthesized. We were expecting some good biological activity, antifungal and antibacterial, but we couldn't found some better activity. Later, our collaborator tested this compound for the quorum sensing. Quorum sensing is basically a process through which the bacteria communicates, and they produce a biofilms which help them in making resistance to certain antibiotics. And the quorum sensing results indicate that most of the compounds, they inhibit the quorum sensing or biofilm formation. If they inhibit the biofilm formation, it means the drugs, they can act on the bacteria. Though these compounds are not acting as antibacterial, but they help in the other drugs to acting as antibacterials. So after clubbing these two nucleus, then we look for the 1,2,3 triazole nucleus to club with the urons. Again, we reviewed the literatures. These are the certain activities associated with the 1,2,3 triazole nucleus. Here, initially we reviewed the literature and then tried to functionalize hydroxy functional groups present in the 6 hydroxyurons to make a triazole nucleus, and then we analyzed to these compounds for the cathepsin B activities. Cathepsin B is having some relationship with the anti-cancer activities. Cathepsin B is a necessary part and it controls many benign processes in the living body, but if it have overexpressions, it leads to certain disorders. And these are some information related to cathepsin B. Based on this cathepsin B activity, we reviewed the literature, and recently we have published that review in International Journal of Biological Macromolecules. And based on that, these are some triazole nucleus, and based on that we synthesized some triazole hybrids with urons. The question is why we selected uron for cathepsin B. When we reviewed the literature, we found that most of the compounds which are having anti-cathepsin B activity, they contain alpha-beta unsaturated carbonyl functional group like chalcones. And if we see the biological history of urons and chalcone, these are the isomer compounds, and if we review the biological synthesis of the flavonoids, they are interconverted to each other. And this uron are also having the alpha-beta unsaturated functional group. So based on that, we explored urons, and here we functionalize this hydroxy group to synthesize with the triazole nucleus. We have synthesized 2 different types of triazoles. This is benzyl linked triazoles, and in another series we synthesize the phenyl linked triazoles. 18 compounds in this series. We use this protocol for the synthesis of the compounds, and initially we synthesize the cathepsin B inhibition potential of these compounds. This is the data. We use aspirin and curcumins for the 2 processes which are controlling the cathepsin B, and we found that some of the compounds are even better potent compared to cathepsin B. These are the compounds which were found better found to have better activity along with this thiophenyl nucleus. I mentioned that cathepsin B inhibition and anti-cancer activity, they are interrelated. It has been found that gastric cancer is because of, in many of the cases, it is because of the overexpression of the cathepsin B. And therefore, we analyze, along with anti-cathepsin B activity, we analyze the anti-cancer activity. And almost similar results were found. Most of the compounds which were having anti-cathepsin B, powerful anti-cathepsin B activity, they are also having the anti-cancer activity against the AGS cancer cell lines. These are some potent molecules. Okay. This is another series where we use phenyl links. So we have synthesized these 2 club. In this series, we have also explored a sustainable approach. We use the ultrasonication method for the synthesis of compounds, and we have a comparison with the conventional and ultrasonication methods. And this is the comparisons. This is the activity for the phenyl linked. Then we compare these 2 types of triazoles. This is phenyl links. These are the potent compounds. This is a comparison. Benzyl links, phenyl links compounds, they were found to have better cathepsin B inhibition compared to the benzyl, maybe because of the flexibility of the benzyl nucleus. So a comparison between, this is a comparison between anti-cancer activity against AGSL and of these two series of compounds. So in conclusion, we have synthesized 4 different series of hybrid urons. Pyrazole clubbed found very potential anti-cancer agents. These chloroquinolines, though they were not found to have antibacterial activities like chloroquinolines, but they were found to have powerful antibiofilm properties. These 2 triazole type of nucleus. So these hybrid compounds we have synthesized. Now, as far as the acknowledgement is concerned, the entire work have been carried out by my 2 students, Bhavna and Gaurav. Gaurav has submitted the thesis, and the financial assistance is provided by Haryana State Council for Science and Innovation Technology, Panchkula. And these are the collaborators. Anti-cancer activity, we contacted Dr. Vilas D. Nasare, Department of Pathology and Cancer Screening, Chittaranjan National Cancer Institute, Kolkata. For antibiofilm, Dr. Nursing and Professor Neera Raghav. So, thank you for patiently listening.
Thank you, Dr. Suresh Kumar. Any questions from the audience? If not, then let us thank the speaker. Thank you very much, Dr. Kumar. And now I invite the next speaker, Dr. Amit N. Gupta, is from JD College of Engineering and Management, Nagpur.
Thanks to give the opportunity to present.
Nobel laureate talk is there.
Okay. Sure. I just started this. My topic is electrical conduction behavior of HBUE1 resin. Here, the resin is synthesized. We know that the polymer, what is the polymer? It is the long chain of the monomers. And this one is the why the polymer is necessary. And next, the present work gives the electrical conducting behavior of the resin by varying the temperature. So here, first the synthesis, this one is the synthesis. We get the monomers of p-hydroxybenzaldehyde, urea and ethylene glycol in the ratio of 1:1:3 in the presence of 20 gram of PPA, polyphosphoric acid. By polycondensation method, in near about the 5 hours, we get the yield of 75%. Now this one is the scheme of the synthesis of here HBUE1 resin will be formed. The conformation of this one, we find out the solubility of this one, number of solvent we use, and here we found the solubility in DMSO and aqueous NaOH. Then elemental analysis, we did the matches of the elemental analysis is near about the same. It gives the somewhat conformation of the presence of the element. Now, determination of the molecular weight, we did the non-aqueous molecular in presence of DMSO by using the KOH. We find out the average molecular weight by multiplying the degree of polymerization with repeating unit. This gives the data of the DP. DP is found out near about the 16 and average molecular weight is 4176 near about this polymer. After this IR, this one is the IR of given compound and the assignments is found like this. Here at medium sharp of this 3572, it gives the NH stretching secondary amide. Here, broad of the phenolic group, it is found, but it is compared by the literature and found this similar to the formation of this one. Here, the phenolic group, it is found in 3433 broad band, 3433. This one is due to the phenolic group. This CO band 1655 sharp aromatic ring, and this one is given the data. Why this all the present is it is matched with the given literature. Next, electronic spectra. In electronic spectra of this compound which is synthesized is found to be N to sigma star, pi to pi star and N to pi star. and the reading, whatever the wavelength is found, it is matches with the literature, and we found this one gives the correct correct information that the moiety of this one aromatic ring and CO amide linkage, and gives the aldehydic group. After this, we did the 1N NMR for this functional group. So here for this compound, we find out this NMR which matches the literature data. 1.2 delta it for the methyl, and 2.46 this one is for the nitrogen and carbon bonding, these two. This aldehydic group 8.3 this for the aldehydic group, and 7.2 CNH, 7.2 this one is the CNH. So it is also structural information matches with the NMR data. After this one is the topic that the electrical conductivity of the given compound. So this we know that the resistivity is R8 divided by L, where the A is the area of the cross section of the pilot. We find the pilot and we use the two probe method here. L is the thickness of the pilot, R is the resistance, and rho is the resistivity. This one is the formula. Sigma is sigma 0 experimental minus E activation energy divided by KT absolute temperature. Here the log of we plot here by log of sigma versus 1 upon T. We find the plot of the negative, so with the negative signs. It has been found the HBUE1 resin shows the semiconducting behavior due to the presence of linear nature of the plot sigma, log of sigma versus 1 upon T. Mention the temperature. It should be due to the hopping mechanism of the conduction due to the loosely bonded pi electrons at unsaturated center of the polymetric matrix. Here by this tabular form, we find out the log of the sigma and 1 upon T. By using this, we plot the graph. Finally, after the calculation, it is found that activation energy of the HBUE1 resin is 1.3809 kilojoule per moles, and it is in the range of 0.126 into 10 to the power 6 to 0.2128 into 10 to the power 6 Siemens per centimeter inverse in given temperature range in Kelvin. It is found this one. And finally, for the application, so it may be it will be play extremely important role. The development of modern electronics, especially in the communication and computer sector, because as the temperature increases, conductance of the material increases as a semiconductor. And this one is the research paper already published, near about 19. Thank you. And this one book is published, and in newspaper, the article related to the polymers is published. Thank you so much.
Thank you, Dr. Amit Gupta.
Thanks.
Now I invite Professor Anand Aswar. Oh, sorry. One person, Dr. S.S. Rangdale, for his oral presentation. And
please stick up to
so you have 7 minutes.
Properties of copolymer resins derived from adipamide and 2-amino-6-nitrobenzethiazole. Here polymer has a very important role. Copolymer is a large molecule composed of repeating structural units, typically connected by a covalent chemical bond. Copolymer have played an important role for the beginning of life. Here various polymers are there: plastics, rubbers, TP, thermoplastics. These are the classifications on the basis of properties. This is the classification of polymer on the design and functions. These are the copolymer random arrangement. There are 2 types. copolymer alternating arrangements, and chain reactant. Here the molecular structure of aramides, aromatic polyamides. Here also these are the phenol formaldehyde resin copolymer, and bakelite networks are here, and this newly synthesized copolymer is derived from 2-amino 6-nitro benzothiazole adipamide formaldehyde. This is in the mole ratio of 2:1:3 in the presence of 2 molar 200 ml HCl, concentrated HCl, as a catalyst at 124 degrees Celsius for 5 hours in an oil bath with occasional shaking, and ensure through a mixing the separated brown copolymer resin was washed with hot water and methanol to remove unreacted monomers. The properly washed resins dried, powdered, and then extracted with a diethyl ether and then petroleum ether to remove the unreacted monomers which might be present along with the A and VAF copolymer. This is also purified by dissolving 8% NaOH and then reprecipitated by 1:1 concentrated hydrochloric acid and distilled water, and by rapid stirring to avoid the lump formation. This yield is 80%, and the newly polymer is characterized by elemental analysis, UV visible, FTIR, and proton NMR spectra. Here the measurements takes place in 4 steps. First we prepare a pellets, then put in the sample holder, and in furnace and heating the sample and the measurement of resistivity. Here by this relation, exponential law, by this relation we measure the resistivity, and with the help of this relation log sigma is equal to log sigma plus minus EA by 2.303 kT. By plotting the graph between the log sigma and 1000 by T, here it is seen that the temperature increases, the conductance also increases, therefore it is classed in a semiconductor. And also here activation energy increases, the lower in exchange energy for the first polymer, and for the fourth polymer it is less than the first, because the skeleton is increases and the activation energy is depends on the pi bond increases, therefore activation energy increases and electrical conductance increases. This is the conclusion, means this is the preparation, and electrical conductivity of each of these polymer resin increases when increase in temperature, hence this copolymer may be ranked as semiconductor. Thank you, thank you, sir.
Will you please show me that the second last slide? Before that, here, this delta E.
Delta E, what do you call that?
Activation energy.
Activation
energy.
And that is 4.3 to 10 raised to minus 10 joule. 10 raised to minus 10.
Kilojoule.
Because that joule is already a very small quantity of energy, and if 10 raised to minus 10, how will I measure it?
Kilojoule, sir.
Please do that correction. Any question? Yeah. So, yeah. Sir, thank you.
Yes.
Well, thank you, Dr. Rangdale. Now, during the last session, we had the invited talk by Professor Anand Aswar, who is present here. So, I'll request Professor Aswar to deliver.
In fact, very few people are there. Still, for the sake of your knowledge, Professor Aswar, he did his MSc, PhD from this department only. He did his PhD with Professor Bhave. And then after that, he joined Amravati University as a lecturer and retired as professor and head of the Department of Chemistry. And he has number of research papers to his credit. Here, number of people, at least 30
Yeah,
students
33.
have been
33.
awarded PhD under his able guidance, and he has organized a lot of conferences, and he has been an invited lecturer.
Sir, don't, no,
Invited
Sir,
speaker for
always,
so many conferences. Thank you very much. Thank you.
Dodge sir is my teacher, is mentor, friends, philosopher also, and we work together, we stay together, and always whenever I am difficulty, I used to discuss Professor Dodge, Principal Dodge. So respected chairman, in fact, really it's honor and pride for a student to talk with the younger colleague, and that too particularly in the same zone where I have spent 6 years, precious 6 years of my life, same wing, यही wing है जिस wing में हमने काम किया है inorganic chemistry का, जहाँ पे मुंशी साहब, भावे साहब, खराज साहब बैठे थे, and with their inspiration, with their motivations, we people could do something also. इसीलिए मुझे बहुत ज़्यादा खुशी है कि हमने हॉल 2 में हमारा सिलेक्शन हो गया है rather than हॉल 1, क्योंकि ये फील होता है ना कि हम हमारे घर में हैं. Though I am working in अमरावती, but I feel pride that हम मैके आए हैं, क्योंकि नागपुर आए, this is my ये ना अमरावती दो just bread and butter, but it is my alma mater, and so always it always feel pride also. So that's why I wrote that I welcome to you all to my alma mater, particular department of chemistry, and I am really overwhelmed to Professor N.K. साहब. N.K. साहब ने भी हमको बहुत सारा गाइड किया. Whenever I used to conduct आविष्कार another activity, so N.K. साहब and everybody helped me. And this is again this Madam Agrawal is also our own student. So we feel very pride and honor, overwhelmed I may say. And Dodge साहब जैसे लोग हो ना, तो it's always source of inspiration, motivations. So anyway, just this is the situation all we are facing, and our Prime Minister also told that we should think about the sustainable development. So this is the situation all we are facing, particularly आगरा, रायपुर, दिल्ली जो है ना, इस एरिया में कि थोड़ा सा हमारा जो पार्ट है ये बड़ा दिक्कत हो जाता है. So this is the just simple topic. Basically I am the inorganic chemist, material chemist, and we could continue our grandfather research, particularly Professor Kharat Saab particularly, and always used to encourage Gajviya Saab, used to encourage do some research in the material science also. And that's why we shifted little bit from pure coordination to the material science also. And we thought that why not simply prepare, we should use this material as a catalyst, their application also. So this is the simple topic which we are using. This is the outline, no need to. So just this is the catalysis we are using. So as all we are aware, some of my young colleagues, unfortunately less number of students. So basically we should cultivate the habit, imbibe the habit, such kinds of thing to motivate what is the role of catalyst and how the activation energy play its role. So this is a very important slide to understand. That is without catalysis, with catalysis conveniently understand. That is when teaching in the class also. So basically we are teaching teacher. In university, we are mostly teacher. So we should take care rather than the research to teaching also. So that teacher is the person who inspire our own student for their research, toward good research also. And we are the supplier for IIT and other peoples also. So this is the fundamental concept. These are the two types of catalysts. All we know the significance of homogeneous, heterogeneous catalysis. And presently, this is the contribution usually made by the homogeneous, heterogeneous catalysis, biocatalysis, because most of the industrial process based on the heterogeneous catalysis also. So this is the fundamental. Berzelius is the, I may say, the father of this catalysis who developed and designed the definition and several industrializations. So this is the person 1779 to 1848 also. So he defined the name catalysis, kata and meanings. So just fundamental things. So there are several kinds of catalysis: organometallic catalysis, transition catalysis, organocatalysis, visible light, nanocatalysis, biocatalysis. Among these several catalysts, we are using the nanocatalysis, particularly material science point of view. So why nanocatalysis instead of conventional catalysis? Again, there are few things one has to consider, that it is a combined advantage of both the homogeneous, heterogeneous. So one can conveniently understand that is homogeneous, heterogeneous, and this is the quasi-homogeneous catalysis. So which is the bridge between these two, and therefore we get these catalysis molecules. So there are several advantages also of this nanocatalysis. So semi-heterogeneous, mimic type, insoluble, enhanced contact, surface area, sustainability, and easy to tailor rings also. So these are the several advantages, and these are the application different by organic reaction, oxidation, reduction, as well as coupling reaction also. So just about this, we are going to just discuss about the silica coated catalyst. So this is silica-based magnetically recoverable catalyst. We have learned homogeneous, heterogeneous, but magnetically recoverable catalyst, which is very important because very easy to separate with the help of external magnet. Just we should attach the external magnet to the beaker or container and separate our product also. So this is the just schematic representations how the silica magnetic core shell supported catalysis, which is highly selective as well as efficient also, which is economical also. So there are several advantages, and these are the applications of this silica coated magnetic nanoparticle which are supported, and these are the different kinds of reactions where the organic or synthetic chemists conveniently use oxidation, hydrogenation, carbon-carbon coupling, asymmetric synthesis, hydration, novel condensation, microwave assisted, and several things. So no need to. So these are the advantage. The catalyst which we have prepared in our simple work, that is recoverable nanocatalyst. The stability is very important, cost effective, very simple. Ferrites are very simple material, easy fabrication, less leaching as well as simple recovery. Leaching is very important as far as catalysis and product isolation is concerned. So these are the main features, no need to bother. So this is the simple work. This is the core moiety which we have used for the preparation of catalyst. Ferrites, most of the students might be aware about the types of ferrite. There are three kinds of ferrites. Simple, that is, I may say the simple ferrite, then reverse, and then mixed metal ferrites also. So inverse phenyl, which we call is usually. So here we have used the core Fe2O3, and then we have dope or prepare the magnesium iron ferrites materials, simple by co-precipitation methods. Basic aim is to synthesis and utilization of this catalyst toward the organic reactions. So this is the catalyst, the sulfuric acid functionalized silica coated magnetic nanoparticles. So this is the preparative methods, schematic representation of this preparation of magnesium iron silica coated sulfonic acid. So this is the flow chart. Very simple to prepare, just simple, it is a ferrites material, and any laboratory, whether it is urban or rural, you can conveniently prepare. No need the exhaustive, I may say, the instrumentation, and you can send your sample to the characterization, either Chandigarh or some other laboratory, and you get the characterization and identify materials also. So simply 1:1 stoichiometry, simple co-precipitators, and then we have this tetraoleum silicates we have added, fluorosulfuric acids, and finally we get the products. This is the detailed method, no need. So we get this kind of catalyst, and simply by keeping the magnet, external magnet, so we can separate our product simply without any filtration, anything also, you get the good compounds. So this ferrites, or I may say the catalyst, sulfonic acid catalyst, we have characterized by the different methods also. So this is the IR spectral characterization, and we have confirmed the presence of various functional group in the catalyst, and formation of catalyst get confirmed with the help of this IR spectra also. This is the power X-ray diffractions, and which indicate also the nano size, and we have compared this data with the help of JCPD file also. So we got a good structure and confirmation also, three different types of pure spinel, then silicon oxides as well as sulfonic acid. So there is slight decrease in the intensity, and this may be due to the presence of silica molecules. So this is the TGA DTA characterization core for these molecules. So first region, which is due to the presence of water molecules, or some, we may say some adsorb solvent associated with the ferrites, and then in the region of 220 to 350 mainly attributed to the loss of this SO3H molecules, and then finally at a higher temperature the complete our ferrites and then silica coated get under decomposition, and the residue is the iron oxides. So this is the same electron spectroscopy for the materials, and we got fortunately a good surface morphology and demonstrate that the formation of nanoparticles as well as spherical with particle size in the range of 75 to 110. Good nanoparticle. This is little bit faint diagram, otherwise it is very good intense also. We confirm this also. This is analysis, element analysis, EDAX spectra, which also confirm the presence of the silica as well as the other group, and element analysis confirm with the theoretical calculation also. So this is the TEM study also. This TEM study also displays the spherical morphology with dark MgFO3 manganese nanoparticles surrounded by the lighter amorphous silica shell about 25 to 30 nanometer thick also. So confirmation with the help of TET. This BET study also we have performed, and this BET study also provide us the information regarding surface area 21.906 meter square per gram with the pore volume of 0.027 cc per gram also, and pore radius is 25. So the confirmation and formation of this metal oxide made by this, and this is a very important histolysis course. We need to perform the histolysis in order to check the hard magnet, soft magnet, which is very important property as far as the magnetic catalysis is concerned. Histolysis corrosivity, which decide the soft or hard magnet also. So we have performed this study with the help of, and these are the application we have used this simple, just I go to directly reaction. So this is the basic reaction we have used for the preparation of the material, multi-compound reaction with the help of this 2-naphthol aldehyde and urea or thiourea, and using this manganese iron oxide silica sulfonic acid as a catalyst. We have optimized the various reaction condition with the help of solvent and different solvent we have checks. We have optimized with the help of effect of concentration of catalyst. Then the temperature also we have studied. Then we have microwave radiation also different, but we have got 350 watt, the very good yield also 97%. Then the different benzooxanes with different substituent, but with the help of the donor, and this is the possible mechanism for the formation of compounds, and the cyclization occurred due to ammonia elimination and lead to the formation of final products. So this is the recycle, that is we can use this catalyst up to 5th cycle also with I may say the 83% of this reusability, same time confirm the formation of catalysis. Comparable study, and our catalyst found very good as compared to other things. This is the spectral characterization with the help of proton NMR, then carbon NMR also we have confirmed the formation of product. This is the mass spectrum study for the compound also, different analysis. We have performed some biological study also of this compound. Further biological study is in progress because we want to detail with the MIC method also. So these are the different value compared with the standard with the bacteria and fungi, gram positive, gram negative also. So just friends, since we are celebrating the year particularly, and 22 and 23 is again one more year of celebration, International Year of Basic Sciences for Sustainable Development, rather than this sustainable also. So this is, all of us should think and unite to save our Mother Earth, preserving its green also. India has to go long way, and therefore save our environment and make it healthy as well as friendly also. So this is the very important message one can carry with us, because most of the time हम लोग भूल जाते हैं. So as a teacher we should take care, and Inki Saab is from the LIT, Lakshminarayan Institute of Technology also. He might have taught his own student and using taking care, but I used to tell my student we should keep in 5 P's in our mind: petrol, power, paper, plastic and potable water. So if we don't take care of this thing, otherwise we have to face lot of troubles, and it is not a sustainable future, because for sustainability we used to optimum, and for think about the future generation. At the same time we used to embrace the 5 R rules in your everyday lives also.
Thank you.
Reuse, reduce, recycle, and this is the message given by our Sant Gadge Baba, where I have served more than 32 years of my service also. The person who never went to any school also, but he develop such kinds of community who always believe towards the educations also, and always tried for the food for hungry, water for thirsty, cloth for the naked, education poor. These are the 10 commandments you gave. You never went any school also, right. And therefore government of India also today giving very importance in Swachh Bharat Abhiyan.
Please conclude.
Just. So this is the last slide. This is my university pictures. So thank you once again for patient hearing. So it is really honor for me to have some kind of discussion with our senior colleague as well as with my teachers, and always one feel pride also. So I am really happy that sectional recorder in our sections. So thank you once again. Thank you ISKA sectional president Ranjana madam.
Thank you for your extensive work and the presentation. And the message is also, which is very relevant in the present context. We are really short of time, and still I will permit if there is a question from among the audience. I will permit.
Okay, thank you, sir. I request the chairperson, Dr. Yanki sir, and Professor RPS Chauhan sir, sectional recorder, to present a memento to Professor Aswar sir.
लाइट लाइट वो बंद कर दीजिए।
Just a minute. I would first like to conclude this session. I thank the organizers for giving me an opportunity to chair this. I thank all the speakers for their beautiful presentation and also the audience. Yeah, for attending.
Now I request Professor Pratibha Agrawal ma'am to propose a vote of thanks.
Thank you, or a vote of thanks. It is a prayer. It cannot be expressed in words. It is a feel. But still, I'm trying. So please excuse me for this. I feel honored to get this opportunity to propose the vote of thanks at the end of the session. I'm thankful to Dr. Ranjana Agrawal ma'am, Dr. Khati sir, Dr. Chauhan sir for providing such a wide platform to all the delegates, researchers and students. I would like to extend vote of thanks to Honorable Vice Chancellor, RTM Nagpur University and all the authorities of Nagpur University for this great interactive session and the platform provided. I am thankful to Professor Dhonge sir and Professor Yankee sir for chairing the session. I am thankful to Aswar sir also and to all the audience which are present over here for the patience hearing. So I am thankful to one and all. So thanks a lot. And I think now there is a Nobel laureate lecture and which we can attend that.